welcome to this video going through the exam questions. We're now on to the protein section, so this starts on question 29. So the question asks you to describe how the structure of llama hemoglobin is likely to be different to that of camel hemoglobin with reference to the 4 levels of protein structure. So it's basically just a question about the 4 levels of protein structure. So we need to know that we've got the primary structure, secondary structure, the tertiary structure, and the quaternary structure. So nice and easy right from the start. We've got our kind of structure of our answer of what we need to include. So we know that what each level of protein structure is, and we've just got to apply it to the question. So it's asking you, oh, sorry, that's just moved. Let's go back down. We know that the primary structure of a protein is the sequence of amino acids, but just make sure that you link it back to the question. So it's asking you, how is the structure of hemoglobin likely to be different in the 2 animals, the llama and the camel? So the primary structure is the sequence of amino acids. So we could say that they're going to have a different primary structure. That's just shorthand for primary. Different primary structure, which means that they've got a different sequence of amino acids. You can see how the way I've written that, I haven't just said the primary structure is the sequence of amino acids. I've answered the question using that information. So the 2 animals will have a different primary structure, which means that they have a different sequence of amino acids. Okay. You could then go on to the secondary structure. So if they've got a different primary structure, then they'll have a different secondary structure. And prove that you know what secondary structure means. So you could say, e.g., the fold or the localized, better if you said localized folding into, remember the 2 shapes, which are the alpha helix and the beta pleated sheet. Okay, so we've kind of linked it back to the question, and we've said that we really understand what the secondary structure is. Remember the bonds involved, so it's good habit when we're talking about protein structure to always do the bonds in each one. So here you could say held by peptide bonds. Amino acids are joined by peptide bonds. Here you could say held by hydrogen bonds. Okay, and then we can go on to the tertiary structure. So the tertiary structure is kind of the biggest one, really. That's the overall 3D shape. And if we've got a different primary structure, then that means we've got R groups in different places. So having a different primary structure means a different sequence of amino acids. So therefore, a different sequence of R groups, which each have different properties and therefore form different bonds, and will make it fold up differently. So having a different primary structure means a different sequence of R groups, which have different properties and therefore form different bonds and affect the overall folding of the protein. So the tertiary structure is the overall folding of the protein, and therefore you'll get a different 3D shape. Okay, remember we always try to mention what the bonds are. So with the bonds here, we're talking ionic, hydrogen, disulfide, and hydrophilic, hydrophobic interactions. All right, so try and mention those in your answer as well. Okay, so it's going to fold up into a different 3D shape. Okay, now they're both going to be hemoglobin. So although the hemoglobin might be slightly different, if it's hemoglobin, they're going to both need 2 alpha, 2 beta. So both hemoglobin. So that's not really going to be altered. Therefore, you can write therefore as 3 dots like that in a triangle shorthand. So both hemoglobin, therefore they will both have 2 alpha and 2 beta subunits. So no impact on the quaternary structure. The quaternary structure means if it's got more than one polypeptide, and hemoglobin's always going to have 4 polypeptides, 2 alpha, 2 beta, so it's not going to affect that. Okay, so hopefully that helps you how you can just incorporate the 4 levels of structure into a question, just trying to keep linking it back. Next one, hemoglobin is a protein that carries oxygen in the blood of all mammals. The structure of hemoglobin can vary slightly between species. Figure 4.1 shows a llama, a relative of the camel. Llamas live, scroll down. Llamas live at high altitudes, camels live at low altitudes. At high altitudes, the partial pressure of oxygen is low. Llama and camel hemoglobin consists of 2 alpha and 2 beta subunits. Each subunit contains a heme group and is able to bind 1 molecule of oxygen. In the beta subunits, 1 amino acid present in the camel hemoglobin is replaced by a different amino acid in the llama hemoglobin. 4.2 shows a dissociation curve for llama hemoglobin and camel hemoglobin. Okay, so here we've got something called a dissociation curve. Now, we haven't come across this yet in our course, but we can work with it and just try and work through the question, and then it will just be more familiar when you get to that bit of the course. So it says, state the partial pressure of oxygen that results in 50% saturation of llama hemoglobin. So all we're really doing, we don't need to understand it too much. We just need to find 50%, which is going to be just here. We read straight across until we get to the relevant line. So this question asked you, double check, llama, 50% saturation in the llama. So we meet the llama line, which is that one. Then you follow it straight down. Use a ruler, please, if you're in an exam. So halfway between there would have been 3, and we're just before there, so something along the lines of 2.8 kilopascals. Okay, so really all you're doing, oops, that's PA. All you're doing is just reading off the graph there. Even though we didn't know about how to read those graphs, we can work it out. Explain why it's important for the survival of the llama that the llama hemoglobin dissociation curve is to the left of the camel hemoglobin dissociation curve. So what it basically means is that any partial pressure, so let's just say, for example, we chose a partial pressure of 6 to look at. If we follow it up from 6, okay, you can see that at that point, the camel is going to be approximately, if we follow it across, approximately just below 80%, let's just say 80%, or let's say 75% saturated with oxygen, 25%. Whereas if we look where it crosses the llama line and go across, we're looking at more like 85%, let's say. Okay, I haven't got a ruler, that's not great. But at any particular partial pressure of oxygen, the llama hemoglobin basically holds more oxygen. The word we use for that is it has a higher affinity for oxygen. And you'll learn to use that word when we come onto this part of the course, but it's really good to get ahead and just have heard the words before we get there. So what you would say is that the llama hemoglobin, you can write hemoglobin as HB for short. The llama hemoglobin has a higher affinity. That means attraction to. It picks up oxygen more easily. Affinity for oxygen. It needs this. So if we refer back to the start of the question, it needs this because it told us that the llama, if we go back up, use the question you're given, the info you're given. Llamas live at high altitudes, it told us. At high altitudes, the partial pressure of oxygen is low. So if the partial pressure of oxygen is really low, it needs to have a high affinity to oxygen in order to bind enough oxygen. Otherwise, it's not going to have enough oxygen for what it needs. Okay, so it needs this because at high altitudes, there's a low partial pressure of oxygen. So in order to be able to get enough oxygen, it needs a higher affinity. Okay, something along those lines. So it's using the information that we've been given. I'm aware some of those words are unfamiliar to you, but it's good practice to apply to unknown scenarios. Okay, next question is about the protein collagen. So collagen is a fibrous protein. So hemoglobin that we've just been looking at was a globular protein, and now we're moving on to collagen, which is a fibrous protein. So globular proteins are sort of round and so therefore they're soluble, whereas fibrous proteins are these long ones and there's no way they're going to hide in amongst water molecules, so they're always going to be insoluble. Okay, so your first property of a fibrous protein that's different from globular is that they are insoluble. It only says state, not explain, so you don't need to explain why it's important or why it's useful or anything. It's literally just state. The second thing is they're much stronger. So we can say it's strong. And thirdly, we can say it's totally unreactive. Okay. They're the only three answers that they accept for that. So just make sure you know those properties of fibrous proteins. Next question, number 30. Which of the types of molecule is shown in the diagram? And you needed to select from these. So hopefully you can identify that that's your amine group, that's your carboxylic acid. And we know that in amino acids, we always have a hydrogen at the top, just that we recognize that. And there's something here that's unknown. You look at that and you think, oh, I haven't really learnt that. What could that be? That's because that's the R group, and the R group is different in each of the 20 amino acids. So hopefully from the rest of the molecule, at least, you can recognize that that molecule is an amino acid. Also, it's obviously in the protein section of this paper. Next one, question 31. Human insulin is a globular protein with a quaternary structure. One insulin molecule has 51 amino acids. Figure 21.1 shows the sequence of amino acids in one molecule of human insulin. Okay, so amino acids are often abbreviated to a 3-letter code. So that's an amino acid, that's an amino acid, that's an amino acid, that's an amino acid, that's an amino acid. The only amino acids you've probably really heard of are glycine, because that's the most the simplest one that we always talk about. And you will have heard of cysteine because when we look at disulfide bonds, that's the only one that contains the sulfur, and therefore that's the only one where you get disulfide bonds. So that bond there, look, between cysteines is going to be disulfide. And you've got another one here between these two cysteines, another disulfide bond there. So the 3-letter code for cysteine is CYS. And you can just see that each amino acid has a 3-letter code for it. So that's a chain of amino acids shown there. The amino acid cysteine, it tells you, is abbreviated to CYS in figure 21.1. The side chain or R group, sorry, not too far, found in cysteine is shown in figure 21.2. Complete figure 21.2 to show the structure of the amino acid. So they've told you that all of this is the R group. We don't need to know R groups, do we? But that is the R group when you've just got to attach the rest on. So there's the R group. Then you needed to just put the carbon in the middle and then know that we've got the amino group on the left, and then you've got the carboxylic acid group on the right and the hydrogen above. Sorry, there wasn't much room to draw on there. It's a bit messy, but you get the idea. So all of that is the R group. You didn't need to draw that circle with the R in it. You just ignore that bit. I was just trying to show you to simplify it. You just needed to draw the rest of the molecule joined on there. Part B, explain how figure 21.1 shows that insulin has a quaternary structure. So if we go back to figure 21.1, quaternary structure means that it's made of more than one polypeptide. So looking at this, you can see we've got one polypeptide going here, along here, around here, so here, that's one polypeptide, and the other polypeptide you can see is there. It'd be better if I just did that in two highlighters, wouldn't it? So we've got one polypeptide there and one polypeptide here. Okay, so it's made of two polypeptides. That's why it proves that it has, or shows that it has a quaternary structure. It has more than one polypeptide chain. Okay, so all you needed to write for that one is quaternary means that it's got made of more than one polypeptide, and the picture shows that insulin is made of two polypeptides. Okay, so you've proved that you know the definition of quaternary and you've explained how it links to the diagram. Okay, and then you could say that the chains are joined together. We know that they're not just two separate polypeptides. We know that they're part of the same molecule because, as we identified earlier when we were reading through it, you can see that they're joined by that disulfide bond just there. Okay, so it's not just two separate polypeptides that happen to be next to each other. They are joined together. That's part of being a quaternary structure, that they're joined together. Okay, so we could say joined by a disulfide bond. They didn't want you to just say bound together with a bond. They wanted you to identify that that was a disulfide bond that was holding them together. Normally we call it, rather than bond, sorry, you should really call it a disulfide bridge is a better word for it. Okay, part C. So part C says insulin is a hormone that regulates blood glucose concentration. People with type 1 diabetes need to inject insulin to reduce their blood glucose concentration as they're unable to produce their own insulin. Diabetics need to inject insulin before every meal as insulin has a short half-life. Enzymes in the liver cells break down insulin, which removes it from the blood. Insulin glargine is a modified version of human insulin that lasts much longer in the blood. Figure 21.3 shows the sequence of amino acid in one molecule of human glargine with the modifications in bold. So let's just identify where those modifications are first of all, just to make sure we've got our eye in to find them. So there they are. Suggest why insulin glargine is long lasting. So why is it long lasting? Why isn't it broken down like the original type of arginine? Because it said enzymes in the liver cells break down insulin, which removes it from the blood. So how come this new molecule that's kind of a variation of it, why is it not getting broken down by these enzymes in the liver? Okay, so we can see which amino acids are changed. So first of all, hopefully you can identify the position of them. So we've got these amino acids that are changed on the end of the chain. So hopefully you identified where they were. But really what we're after is that why wouldn't an enzyme break it down? Think about what do you know about how enzymes work. So hopefully we can remember that enzymes work because they have this little area here called the active site, and the shape of that active site has to be complementary to the shape of the substrate. So what's going to change the shape of the substrate? What is the substrate? So this is our enzyme that it talked about in the question, and the substrate that's going to fit in there is this insulin, or this modified insulin molecule. Okay, so normal insulin fits in just fine. Modified insulin doesn't get broken down. Why doesn't it get broken down? It must be the wrong shape. Yeah, so it must have changed shape, and therefore it's not complementary Therefore, it doesn't fit in the active site, and therefore it can't be broken down by this enzyme that they're telling us about in the liver. Okay, so for your answer, things that they're looking for is, do you know why the 3D shape of this molecule here would have got changed? Okay, so you're not going to need to talk about your primary, secondary, tertiary, quaternary structure. You're going to need to talk about your levels of protein structure. So it started off with a clue because it told us that some of the amino acids had changed. So link it to protein structure. So if amino acids are changed, link it to the question. They told you that amino acids were changed, link it to your knowledge. So amino acids have changed, therefore the primary structure of the protein has changed. Okay, so link it with the words that they used in the question to the words that you have learned about the structure of protein. So because the amino acid structure has changed, therefore the primary structure of the protein has changed. Okay, you can then say that the tertiary structure of the protein will be changed because the primary structure always affects the tertiary structure, because the R groups will be in different positions. Remember, the R groups have got different properties, and therefore you'll get different bonds forming. Bonds will form in different places, and therefore it folds into a different 3D shape. Okay, so we're proving now that we understand the link to tertiary structure. We're proving that we know that tertiary structure means the overall folding, the overall 3D shape. We're showing off our knowledge. So because the primary structure's changed, therefore the tertiary structure's changed, and you explained the link to primary. So if you've got a different order of amino acids, we've got different R groups in different positions, different bonds will form in different places, it's going to fold up into a different 3D shape. Now link it to the question. It's asking you, why will this modified insulin molecule not be broken down by the enzyme? So if it's a different shape, it won't be complementary. It won't have a complementary shape to the active site of the enzyme. Where's that gone? Can't find it. Of the enzyme. Okay, so if the shape of this substrate, now it's been modified, as folded into a shape where it no longer fits, where it's no longer complementary to the active site, then you're less likely to get enzyme substrate complexes. Okay, and therefore it's less likely to be broken down. Okay, now this question, strangely, was worth 1 mark, and you only needed to say one element of that. But equally, that same question could easily have been worth 6 marks. So don't feel like it's a waste of time going through the full detail of how that would work, because absolutely another time it could be worth way more marks. All right. All we're doing is practicing and trying to prep ourselves for any possible future exam questions. So the more detail we can put at this stage, the better. Part 2, the table shows some of the DNA triplets for amino acids. So hopefully you've seen a few different grids. Generally, we get one that's like in a table like this, in a sort of square rectangle shape, and we get another one that's in a circle. Make sure you're kind of familiar with how to read them. You obviously don't need to memorize them. So it says, in order to produce the insulin glargine, the human insulin gene is modified by genetic engineering. This is a process that can change the genetic code of the gene. The genetic code of the triplet, DNA triplet 21, is changed, so the amino acid codes for the new version, codes for glycine instead of asparagine. With reference to the table, predict how the genetic code of triplet 21 is changed so that it codes for the amino acid glycine instead of asparagine. So we need to find those on here. Now, remember the genetic code is what we call degenerate. So that means that some amino acids are coded for by more than one triplet. So we're looking for asparagine, which is just here, okay, and glycine, which is just here. So, first of all, asparagine, this table shows us that asparagine, actually, let's leave it in yellow, can be coded for by this triplet or this triplet. Okay, asparagine isn't anywhere else on that table, so it's just those two that we're looking at. It hasn't given you the whole table, look, it's only given you the first base A and G. It's just a section of the table. Glycine, on the other hand, let's just go over that purple, can be coded for because it's in the middle of that box. It means that it can actually be coded for by this triplet, this triplet, this triplet, or this triplet. So there's 4 possibilities. Now, if it's just had one mutation, what we're looking for is what could that mutation have been. So it's obviously started off as asparagine. If you look back down to the question, I forgot. I just wanted to double check which way round it was. I forgot. So originally, so it's changed. So the amino acid it codes for is glycine instead of asparagine. Okay, so originally, just write it at the side so we can see the table at the same time. So originally it was asparagine. So originally it was AAT or AAC, and now it's changed to glycine. So now it's either GGT, GGC, GGA, or GGG. Okay, so it was that, and it's changed to be this one. So if you literally just said that this or this has been replaced by one of these, that automatically gets you the 2 marks. So 1 for saying that originally it was one of these, and now it's one of these. That's 1 mark for that, 1 mark for that. However, you might have worked out actually how it changed. So you could have said, for example, that it might have changed from that to that, and the 2 A's were replaced by 2 G's. Yeah. You could have said, for example, that maybe it changed from that one to that one, in which case you'd say those 2 A's there got replaced by those 2 G's there. Okay, it doesn't matter how you describe the change, and actually didn't need to describe the change in that particular question. You need to say identify the triplets it was originally and that it changed to one of those triplets. That's all you needed for that. Part 3, the modified polypeptides that form insulin glargine are made inside cells. The process of making the modified polypeptides that form insulin glargine involves several steps. The process starts with the modified gene for insulin glargine. Outline the steps involved in the making of modified polypeptides. So it's asking you, how do we make a protein from a gene, starting with the gene until the modified polypeptides are made. So this is a big question. It's asking you to go all the way from the DNA right through to where a protein is made. Now, you've only got 4 marks to do the whole of transcription, translation, and then modification in the Golgi. So you're not going to be expected to say every single step. We're going to just need an outline. So first of all, the DNA, which is obviously where the gene is, gene's part of the DNA, isn't it, starts off in the nucleus. Okay, then you could say in the nucleus it makes an mRNA copy of the gene and name the process. So that process is called transcription. Then you could say that the mRNA gets modified to produce the mature mRNA. We could call that splicing. Then we can say that the mRNA leaves the nucleus and goes to the ribosome. Goes to the ribosome. You can write this in sentences. I'm just trying to save time, writing it shorter. So then at the ribosome, we're going to make the actual polypeptide. So the mRNA is used as a template to make a polypeptide. So that process is called translation. You could just put like a tiny bit about translation, something like the tRNAs bring the amino acids. So you could talk about the codons, anticodons binding, complementary ones. Oops, anticodons. You could probably say specific amino acids rather than just amino acids. Okay, so the correct codon on the mRNA binds to the complementary anticodon on the tRNA, and the tRNA then carries the one with that anticodon will be carrying a specific amino acid, and then your amino acids are going to bind together with peptide bonds. And then the final step is it says it needs to be modified, and we know that modification and packaging of a protein happens in the Golgi. All right, it's only a 4-mark question, so you're not going to be able to go into all the steps of transcription and all the steps of translation. It's just going to be the sort of overview as it goes through the cell. Number 32, which statement about the secondary structure of a protein is correct? So secondary structure is your localized folding, and that includes alpha helix and beta pleated sheet. Make sure you know roughly what they look like. Like that. So beta pleated sheets contain beta glucose. Well, no, because that's a carbohydrate and this is a protein. Disulfide bonds are not involved. Now that's right, isn't it? Because disulfide bonds are in the tertiary structure. Each single polypeptide is either an alpha helix or a beta sheet. That's not true. If we got one long polypeptide like this, and it was going to go through its secondary structure folding, it might be that you get a little bit of alpha helix. It might be that you get a little bit of beta pleated sheet. Okay, you might get another bit of beta pleated sheet. Might get another bit of alpha helix. So one polypeptide can have areas of both types of folding. So it's not that a single polypeptide needs to be all alpha helix or all beta pleated sheet. Okay, that's not how it is. It can be like this, have elements of both types. So that's not right. And hydrogen bonds form between the R groups of different amino acids. Now, we know that the secondary structure is held between R groups. Okay. Sorry, we know that the secondary structure is held with hydrogen bonds. Okay, but they don't need to be different amino acids. Okay, so we go with B. Disulfide bonds are not involved. Oh, sorry. I was just thinking that D. Does that make sense? The thing that's different about, wrong about it, is that it's not between R groups. The hydrogen bonds don't form between the R groups. They form between other parts of the amino acid. That's where that is. Yeah, that's right. Right, number 33, hemoglobin is globular protein. 16.1 shows the structure of hemoglobin. Now, obviously, your exams are in black and white, but we know that within this structure, somewhere there are 4 subunits because we've learned that. And you can see that each subunit has this structure in it. So that'll be your non-protein prosthetic heme group. So there's one of those in each of the 4 polypeptides. Okay, so you can see that element. It's hard to see the 4 polypeptides because it's printed in black and white, but it will always be printed in black and white. There are 4 levels of protein structure. Name the bond present in the primary structure of a protein. So we've talked through these a few times, how we need to know the bonds. So primary structure, the amino acids are held together with peptide bonds. Name the structure labeled X. So it's pointing to the little bits within the polypeptide that are non-protein. You can see that they're not protein. You can see that they look different. So those prosthetic groups are called heme groups. Usually we spell it H-A-E-M. You can spell H-E-M-E. I think that's American. I think that's why you sometimes see it like that. The less specific prosthetic group in that case doesn't give you the mark, although it is a prosthetic group, it's just not specific enough. You're meant to know hemoglobin really well, so you're meant to know it's a heme group. State one feature visible in 16.1 that shows that it's a globular protein. So globular protein is basically just your shape. So you can see that the molecule is spherical, as opposed to like an elongated long fibrous shape. You can see the shape is spherical. It's not got any fibers. Another thing, it's got prosthetic groups. Only globular proteins have prosthetic groups. So the fact that it's got the prosthetic group shows us that it's globular protein as well. But the main thing is spherical. 34, figure 5.1 shows a part of a conjugated protein that's a respiratory pigment in muscle cells. Which part of the molecule does 5.1 represent? Okay, so we should have options. Sorry, let's go down a little bit. So is it a prosthetic group? Is it a disulfide bond? Is it a quaternary structure? Is it a polypeptide? So a polypeptide is a chain of amino acids that start at the bottom. Is that a chain of amino acids? A quaternary structure means that it's more than one polypeptide. Is that more than one polypeptide? Disulfide bond is going to be sulfur to sulfur. Okay, disulfide to sulfur. And a prosthetic group is something that's non-protein. Okay, so if we look at the molecule again, let's see what you can see. So going from the bottom, the options are, is it a chain of amino acids? We know amino acids, we know the COOH group and an NH2 group. Have we got those? No, we haven't. It's not a chain of amino acids, is it? It doesn't look like a chain of anything. Is it more than one polypeptides? Do they look like polypeptides to you? Now, where have we got these like corners or junctions on molecules like that? Each of these is going to be carbon. Okay. It's just been simplified in the way that it's drawn. So no, that's not a polypeptide chain, is it? Or it's not more than one polypeptide chain. Is it a disulfide bond? Have we got sulfurs in there? No. Is it a prosthetic group? Is it non-protein? Yes, it is. It's not protein. It's not amino acids. In fact, we can see that it's got the iron in the middle there. And we know that iron is not part of amino acids. All right. So it has to be a prosthetic group, a non-protein group there. And then the next one, a conjugated protein is held together by many different types of bond. Which bond is not formed when a conjugated protein folds into its quaternary structure? So we're looking really at kind of 3D overall shapes. Now, the bonds we've learned, we didn't really talk about, we don't normally talk about bonds in quaternary structures. We normally talk about the bonds in the tertiary structures, but it kind of means the same kind of thing, and it's folding up. So in terms of its folding, it specifically says what bonds are formed when it folds. Disulfide, yes. Hydrogen, yes. Ionic, yes. Peptide, no. It does contain peptide bonds between the amino acids, but they're not involved in the folding of it. They're just involved in making it into polypeptide in the first place, joining the amino acids together. Okay, next one. Lipoproteins contain protein, cholesterol, triglycerides, and phospholipids. They're involved in transporting cholesterol in the blood. The protein molecules in a lipoprotein are made of amino acids. One of the amino acids is alanine. The R group of alanine is CH3. Describe the structure of alanine. Right, it's given you a space. If you've got a space, you can use it. You can do a drawing before you do your description. So we know amino acids are carbon, hydrogen, R group, amino group, and carboxylic acid. Yeah, we know that off by heart. So you can always draw that. You can always label it. So you could label that that's your amine group. You could label that that's your carboxylic acid. Now, instead of writing R there, it's told us that the R group is CH3. So instead of putting R, put CH3 there instead. Okay, so that bond would join onto the carbon. Do it straight down. All right. So instead of the R, that's what our R group is. And then you can describe it. So you can start off by saying there's a central carbon. You can say it's got an H above, better rather than above, because it could be in any orientation. You could say attached to the carbon. You could say that it's got an amine group. Prove that you know what an amine group is, NH2, attached to the central carbon. And then it's got a carboxylic acid, COOH, attached to the carbon. Okay, so you can get a good description, but if you'd made a mistake in your description or not been detailed enough, if you've got your drawing to back you up, it's more likely to pick up the mark. So if you've got a space for a little drawing, add a little drawing in. There's no harm in doing it, and it's really likely to add to your marks. Okay, so don't be shy of doing drawings. You're never going to lose marks. 37. Pepsin is an enzyme that digests protein foods in the mammalian stomach. Protein molecules are made from chains of amino acid. Name the covalent bond between amino acids in a chain of amino acids. Obviously, peptide bond. Name the type of reaction involved in breaking the bond. So when you make the bond, it's condensation. When you break it, it's going to be hydrolysis. So name the type of reaction, hydrolysis reaction, and describe what happens in this reaction. So we know that condensation reactions make a molecule of water. Hydrolysis is the opposite, so they use a molecule of water. Okay, so what condensation reactions make water, hydrolysis reactions use water. Okay. So that moves us on to the last section, which is amino acid chromatography, which I'm just going to do in a separate video in a second. Okay.